Hi, physiology students, and welcome to this workshop on cellular metabolism. Hopefully this video will give you a good idea and summary of what cellular metabolism is as you're working through your handout, as well as give you some extra practice and understanding into the topic of cellular metabolism for your physiology course. So what is cellular metabolism? It's also known as cellular respiration, and it's the way your body's cells take in glucose from stored energy sources or directly from the food you eat and turn it into energy. And energy in your body is ATP, adenosine triphosphate. You need ATP for many different chemical reactions in the body. Your body needs energy to survive, and it needs to create energy as ATP, adenosine triphosphate. Adenosine triphosphate has so much energy in the bonds that are between the three phosphate groups. And when those bonds break, they give off energy. And that's why ATP is so important. So we get energy by eating or by using fat stores. And our body can turn glucose into energy using three simple steps. They're a little complicated, as you can see, as if you're trying to fill out your handout, but they're also really important for understanding. And again, this video will hopefully take you through those three steps briefly and simply. So the first step of cellular respiration or cellular metabolism, I'll use those words interchangeably, is glycolysis. And glycolysis literally means splitting a sugar, glucose, into two parts. This always occurs in the cytosol of the cell, and it breaks down each glucose molecule into two molecules of pyruvic acid. That pyruvic acid then goes into step 2 of our cellular metabolism. Pyruvic acid enters into the mitochondria now, specifically into the mitochondrial matrix, where the citric acid cycle or the Krebs cycle occurs. During glycolysis and the citric acid cycle, we do what we call substrate level phosphorylation, which forms very small amounts of ATP. And then the third step of cellular metabolism is called oxidative phosphorylation, or the electron transport chain. And this is where energy-rich electrons that have been picked up by coenzymes from the previous two steps are now transferred to the electron transport chain. This chain is built into the inner mitochondrial membrane of the mitochondria, and the electron transport chain will carry out what we call oxidative phosphorylation. And this is what generates most of the ATP, about 30 ATP molecules just in this one step in cellular respiration. So those are the three steps which turn glucose into energy. This is cellular respiration. And now we'll go over these three steps a little bit more in detail. And feel free to pause this lecture video as you're filling out your handout, because some of these pictures will show you the names and give you direction in how to fill out the specific steps in your handout as well. So phase 1 of glycolysis. So let's talk about glycolysis first. We're in the first step of cellular respiration. What we're going to do is we're going to split a glucose molecule into two pieces. But we do that in 3 different phases. And this is phase 1, called sugar activation. What we're doing is we're phosphorylating or activating the glucose by adding on 2 phosphate groups to either side of the glucose molecule. So your glucose molecule is made up of 6 carbon atoms, and each little circle stands for a carbon atom. Phosphorylation will add 2 phosphate groups to either side of the glucose molecule so that we form this molecule, fructose 1,6-biphosphate or bisphosphate. I'm a chemistry nerd. It was my major in undergrad, so I love the language of chemistry. What we have here is we've added on phosphate groups to the 1 carbon and the 6 carbon. So that's why it's called fructose 1,6-bisphosphate. And to do this step, we actually need 2 ATP in this first step. And you're probably thinking, we're trying to make ATP, not use it up. But this first phase 1 of glycolysis actually requires 2 ATP to do this phosphorylation step. So this slide will help you hopefully fill out that first handout where you're filling out some of the labeling. Phase 2 of glycolysis is what we call sugar cleavage. So now we'll actually take this molecule and we're going to cut it in half or cleave it. So we take the fructose 1,6-bisphosphate and we're going to split it into 2 3-carbon fragments. So you can see here the 3 carbon circles still attached to their phosphate group, and they each get a name: dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And again, pause the lecture video because this step will directly help you fill out the next kind of step of glycolysis in your handout. Phase 3 of glycolysis is the sugar oxidation and ATP formation. Whenever you see the word oxidation, it means we're taking away hydrogens and transferring them to something else. So that's oxidation, taking hydrogens away. If you ever hear the word reduction, we're adding hydrogens onto a molecule. But sugar oxidation, what we're doing here to each of these 3 carbon molecules is we're actually going to take hydrogens away and transfer them to an NAD molecule. So what we're going to do is we have 2 NADs here standing by. The positive means they're positively charged, and we are going to transfer hydrogen ions to them to form NADH, and we'll get 2 of those. And these 2 NADH molecules are going to hold these electrons and with those hydrogen bonds to them. And these are called high energy electron molecules, and these will be very important for transferring and using their electrons in the final stage of cellular respiration called the electron transfer chain. So in this step, we form 4 ATPs as well, and this is our last phase of glycolysis. So one of your application questions has to do with the amount of ATP or the net ATP that's formed from glycolysis. We form 4 ATP from glycolysis, but remember we had to start with 2 at the beginning of phase 1. So the total or the net ATP that we're actually forming is only 2 because we had to start with 2 and we get 4 out at the end. So net gain ATP is only 2. So what we're doing here is we're forming 2 pyruvic acids, which will go on and enter our second step of cellular respiration, to the citric acid cycle. This is in the presence of oxygen. The 2 pyruvic acids will go on to the citric acid cycle in the presence of oxygen. And without oxygen present, pyruvic acids are actually converted to lactic acid. And lactic acid can cause kind of the burning sensation when you've been working out a lot or running for many miles, maybe some of you have run marathons before. So this buildup of lactic acid occurs in the absence of oxygen. So pyruvic acid will get converted to the lactic acid and contribute to the increased acidity. And that lactic acid is the burn that you feel if you've been working out for long periods of time and your muscles have depleted their oxygen storage. So let's go into step 2 of cellular metabolism. This is the citric acid cycle. And first, before we kind of enter the citric acid cycle, we're going to go through a little transitional phase. Sometimes it's its own separate step, but often you'll see it kind of combined into the citric acid cycle. And this is how your handout looks as well. It has this transitional phase at the top, and it's taking the pyruvic acid from glycolysis and turning them into acetyl-CoA. So what it's doing is it's oxidizing the pyruvic acid and turning it into acetyl-CoA, which will then go on and enter the citric acid cycle. In this transitional phase, we're forming carbon dioxide as a waste product, and we're forming another NADH, which is again our high energy electrons that are being transferred in the form of NADH. You'll see carbon dioxide several times given off as a waste product, especially in this Krebs cycle. We see a carbon dioxide here as a waste product, and then we see 2 more carbon dioxides here as a waste product. And again, you'll need to kind of probably pause this lecture video to kind of use this as you're filling out the handout as I talk through the different steps of this cycle. So the carbon dioxide as a waste product is waste from this idea that your cells are making energy from glucose. Think for a minute how your body gets rid of this carbon dioxide waste, and exhaling is one way that our body gets rid of CO2. So we formed acetyl-CoA in this transitional phase, which has 2 carbon atoms to it. So again, you can kind of see these carbon atoms will be reduced or will be oxidized as we're going around the cycle. So we often start with oxaloacetic acid because this will be the pickup molecule, and we kind of start and end the citric acid cycle with this molecule, oxaloacetic acid. What that will do is this is a 4-carbon molecule. It will combine with acetyl-CoA, which has 2 carbons, to form citric acid in the initial reactant. You can see citric acid has 1, 2, 3, 4, 5, 6 carbon atoms. It will be kind of changed or transformed into isocitric acid, still with 6 carbon atoms. And then we're going to oxidize and as we go around the cycle. And as we do that, we will get rid of a carbon, usually in the form of carbon dioxide, and those excess electrons or hydrogens will be transferred to NAD to form NADH. And we'll do that several times. You can see that we form 3 NADHs as the cycle goes around, as our carbon molecule is oxidized, because again, we're taking electrons away. We form carbon dioxide several times as a waste product. We'll form a few ATP at the end, but the cycle will continue to go around. It will also form FADH2, which is another molecule that holds these high energy bound electrons in the bonds with the hydrogens that it picked up from one of these oxidized molecules. And you can see here, we just form alpha ketoglutaric acid, succinyl-CoA, succinic acid, fumaric acid, and malic acid. And again, kind of big picture, take a step back here. You know, we start with oxaloacetic acid, 4 carbons. We combine it with a 2-carbon acetyl-CoA to get our citric acid, which is what we start with, our 6-carbon initial reactant. And from here, the cycle will go on by taking away electrons in the form of hydrogens and transferring them to NAD to form NADH or FADH2, as well as forming carbon dioxide as a waste product and a few ATP at the end. So that is the citric acid cycle, briefly summarized to hopefully help you fill out your handout and give you a better kind of look at this step in cellular respiration. So I'm just going to go back real quick here. If you remember from glycolysis, we get 2 pyruvic acids. So we will get 2 acetyl-CoAs. So this cycle will actually go around twice for each glucose molecule that we split. So for each turn of the cycle, we get 2 carbon dioxides, 4 molecules of reduced enzymes, which are our 3 NADHs, our FAD, and 1 ATP. But remember that we started with 2 pyruvic acid molecules. So what that means is the cycle just goes around twice and our final products from 1 glucose are doubled. So from 1 glucose, we get 4 CO2s, 6 NADHs, 2 FADH2s, and 2 ATPs. And when we add those to the transitional phase, this is kind of our final breakdown of 1 glucose, and our final products using the transitional phase and the Krebs cycle. These are our final products from that second step of the citric acid cycle. All right, so if you're still with me, good work. The final step of cellular respiration, again, the way our cells take glucose and turn it into ATP, is the electron transport chain and oxidative phosphorylation. And this occurs within the inner mitochondrial membrane of the mitochondria. The electron transport chain will create what we call a proton gradient across the inner mitochondrial membrane using high energy electrons removed from food fuels. And chemiosmosis uses the energy of the proton gradient to actually synthesize and make ATP. And this is where the majority of our ATP is formed. Again, just using 1 glucose molecule, we're trying to create as much ATP as we can, and the majority of the ATP is formed in this step. So here is a look at the electron transport chain. It's a really fun, interesting step to look at. You can see here the intermitochondrial membrane where my laser is. The intermembrane space is above it, and the outer mitochondrial membrane will kind of be above here. You can kind of see the outer mitochondrial membrane there. Remember, your mitochondria have 2 membranes within them, and the inner mitochondrial membrane is where we have these kind of protein carriers. They're Roman numerals 1, 2, 3, 4, and 5. And number 5, the protein kind of channel is called ATP synthase, which is actually like a motorized rotor, which will spin. And we'll talk about how that works. So phase 1 is where we have the electron transport chain creating a protein gradient. And how that happens, we take our reduced coenzymes, which are NADH and FADH2. They're reduced because they have these extra electrons in their bonds with their extra hydrogen atoms that they picked up. These will deliver their electrons to respiratory enzyme complexes 1 and 2. And what they'll do is they're going to pump their electrons through these complexes. And as the electrons are kind of pumped through the complexes, those are shown in the yellow pathway arrow, hydrogen ions are pumped up into the intermembrane space to try to create a gradient of lots of hydrogen ions up here in this intermembrane space. Q stands for a coenzyme ubiquinome, and cytochrome C stands for another kind of coenzyme, which helps to shuttle electrons between these larger complexes. And in your handout, you need to label this Q. It's lipid soluble, so you'll find it in the lipid membrane, and cytochrome C is not lipid soluble, so you'll find it on the exterior side of the membrane. A little fact about cytochrome C, some toxins and carbon monoxide, for example, will inhibit cytochrome C, which will directly affect this electron transport chain. So some toxins, carbon monoxide will inhibit cytochrome C and will lead to a decreased production of ATP. So again, what we're doing is we're transferring electrons through these complexes, and you can follow their pathway in yellow. And when we transfer those electrons, we're pumping hydrogen ions across to create a gradient. Chemiosmosis uses that protein gradient then to synthesize ATP in step 2 or phase 2. So once we create this gradient of hydrogen ions up here in the intermembrane space, our ATP synthase kind of comes alive, and this is where the magic happens. When the ATP synthase starts to turn or rotate its motor, the hydrogen ions will rush through the ATP synthase molecule, and the rushing through of those hydrogen ions down its electrochemical gradient will rotate this motor. That rushing down will kind of like start a conveyor belt action of creating ATP. This ATP synthase, or complex Roman numeral number 5, will harness the energy of the proton gradient. As hydrogen ions flow back across the membrane, the synthase rotor will spin, causing a phosphate group to attach to ADP, forming many, many molecules of adenosine triphosphate. The last kind of step that I haven't talked about yet is what happens to all of these excess electrons that are being transferred through. They can't just hang out in the mitochondria forever. What they do is they will combine with hydrogen and oxygen to form water. And this is where you will learn why your body needs oxygen to survive. Oxygen is what we call the final electron acceptor, because oxygen will combine with these excess electrons. Adding in hydrogen, we will form water, and water is another waste product from cellular respiration. So again, if you need to pause this lecture video, this will hopefully help you answer and fill in a lot of the blanks to fill out your handout. I have a couple of final slides about the ATP synthase kind of rotor molecule. It's fun to see, again, how this rotor will turn as hydrogen ions rush through to create those ATP molecules. And then a final picture, which is just really fun to look at. This is an atomic force microscopy image that's actually revealing the structure of those ATP synthase rotor rings. So if you go back to this picture, you can see that it looks like a rotor from a machine. And if we actually zoom in on the inner mitochondrial membrane, this is what ATP synthase looks like. And you can kind of see an individual rotor molecule there, which is just kind of fun to look at. So I hope this video helps you to fill out your final or your handout for this workshop. If you can attend a live session, the instructor will help even more, or ask the MDSC for a tutor. Thanks for listening, guys. I hope you're all doing well and take care.